short and sweet one, hopefully. Don't have any announcements from last lecture. Any questions before diving into this one? Follow up from last time. Okay. So as I said, synaptic transmission, we're going to focus a bit on anatomy of neurons, talk about how those neurons function, why it's important to what we're studying. At the end of the lecture, go over agents that alter neuron functioning, and that will be important when we're talking about experimentation with the drugs that we're talking about. Next lecture is describing the specific neurotransmitters that are important. And at the end of that lecture, I will cover the in vivo experimentation that's done for testing drugs of abuse and that. So, and those experiments can sometimes employ these agents that alter neuron function. So, just where we're going. Okay. Nerves, or neurons, are highly specialized cells that we have in our nervous system. And the basic breakdown is the cell body is here, and in the neuron world. In neurons, cell body is known as the soma. It has all of the standard membrane-bound organelles that everybody learned about in our basic and cell biology courses. So they have all the same functioning and energetic requirements in that as normal cells, but then they have a highly specialized function that is derived from the other apparatus or apparati. Here we see these tree-like projections known as dendrites that come off of the soma, and then usually the longest one is known as an axon. At the end of the axon, they also have some projection here. These are the terminals of the axon here. Some of the certain neurons also have one other added feature, are these myelin sheaths here. Proteinaceous and fatty structures that will wrap around these axons and allow for quicker transmission of messages and impulse from the soma out to the terminal end, and why that's important we'll get into. Now, as I'm sure many of you know, these don't just exist in isolation in our body. There's long chains of these neurons all the way from our brains to the tips of all of our extremities. And these cells are lined up together, never touching, but they have these, so a dendrite of a neighboring neuron will butt up against the terminal of the axon of a neighboring nerve cell, and all of the magic happens here in this space that we know to be called the synapse, right? In this synapse, we have release of neurotransmitter out into the synapse, and that's how they communicate with each other. So here we have presynaptic nerve terminal, that would be this neuron here, and then postsynaptic terminal, that would be this dendrite, and then a neuron here, right? These synaptic vesicles will release neurotransmitter out into the synapse, and that's where it binds to its receptor and does all the magic that we've been talking about. Most of these receptors and parts of the neuron are targets of the drugs that we'll be talking about. So the functioning of neuron is very important, because not only is it the target of the drug, but how the neuron functions, will be usually amplified by illicit drug use, and that causes the effect of the drug. So, but basic flow here, you have neurons, sequence. Once they're triggered to, they will presynaptic nerve terminal releases neurotransmitter out into the synapse that can then bind to a waiting receptor on the membrane of the postsynaptic nerve terminal, and that is how neurons communicate. So, what is also important is that, again, it functions like a normal cell, has a nucleus, the endoplasmic reticulum, Golgi apparatus that are all done for processing polypeptides. After translation of a transcript from the nucleus, it'll get modified by these structures, and then other macromolecules that are important for neurotransmitter development will be transported down the axon using these microtubules in a anterograde transport type situation. And then down here, the terminal is where everything, neurotransmitter and whatnot, gets packaged into these synaptic vesicles, these membrane-bound neurotransmitter-containing things. And those are allowed to bind to the membrane and release neurotransmitter outside the cell. Okay? Hopefully very straightforward. Now, what's really important to remember is that this neurotransmitter release happens in conjunction with the electrochemical potentiation or a change in the electrochemical potential of a cell, right? These two things are linked together and happen in conjunction. It's not just one or the other, they both have to happen together. So it's important to remember that when studying neurotransmitter release. Again, as just stated, these are a chain of neurons from the central nervous system out into the periphery. So we have the axon terminal of one, synapsing on the dendrites of a neighboring neuron, and then so on and so on for as many as are in that chain. But again, what's important is that prior to neurotransmitter release, what has to happen is a string of action potentials all the way from the soma out through the axon down to the end of the terminal. Action potential can be recorded like this. So membrane potential of a neuron is shown in the y-axis here, x-axis is time, and a quick bottom line here is that due to the presence of positive and negative ions and species that carry those charges within the cell, the overall charge of a neuron is negative, well, most cells, I guess, but yeah, and we'll get into this in just a minute, but the idea is this action potential is a quick change from that resting membrane potential, and then all of that quick change and displacement of negative ions causes the cell to be able to do work, essentially. And this will then allow for neurotransmitter release, as we'll say in a minute. Back to here, we can record, or we can record the change in this potential and see how this affects the functioning of a neuron. More specifically, we have a neuron in a lab, on a slide, okay? And if we put a recording device into that neuron that acts as a voltometer, or that can read charge of a cell, these cells have a negative resting potential, right? So they have an overabundance of negatively charged things, usually ions and proteins and nucleic acids, that cause the overall charge of the cell to be negative. One side of the membrane is negative. Usually, so negative 60 millivolts is a common resting potential for neurons, usually anywhere from negative 40-ish to 90 to 100, negative 90 to 100 millivolts is the usual range for these. We could just pick negative 60, it's very common. Depends on the neuron, depends on what's in a number of things, but the bottom line is it sits at a negative charge while resting, all right? So look at that a little bit further, or how this happens. So this is a cross-section of a neuron, all right? So imagine cutting a neuron right here, and then looking end-on into that axon would look something like this. So again, we have our resting negative potential here, caused by negative ions on the inside and positive ions are on the inside and outside, but there's an abundance of negatively charged species inside, mostly ions. The way the cell is able to do that are through ion pumps and channels. One example here, the sodium-potassium pump. So this is an ATP-dependent process whereby this pump can import potassium ions and export sodium ions. Seen here, other ports like this chloride pump can just pump chloride outside, all right? So we have things going in and out or just out depending on the type of ion and the needs of the cell. And then channels, as we talked about before with receptors, these are just a pore that allows for specific ions to move across the membrane. And again, it'll move down its membrane potential. From high concentration to low concentration. Hey, I did that again. Sorry, buddy. My apologies. Okay. A bit more specifics. You don't need to memorize these numbers by any means, but just showing some of the more important ions in a cell: potassium, sodium, chloride, calcium. Potassium is highest inside the cell. So values here are intracellular values. These are extracellular values. They are listed in millimolar. Potassium is highest inside the cell. Sodium, chloride, and calcium are highest outside the cell. All right? So this just describes the makeup of a neuron, right? And why it has a negative resting membrane potential. What is important to note is that these will establish a certain equilibrium at resting potential. Even at resting potential, though, potassium is the only ion that can leak from a cell. When it's at a resting membrane equilibrium, potassium or sodium and chloride usually don't flux, even though they can be pumped across in an energy-dependent manner. Potassium really is the only one that kind of leaks. For the most part, it kind of locks down once it's at its resting potential. But potassium is able to sort of, it'll move from inside to outside, again, based off of concentration, but it's more of a very tiny leak that doesn't change that a lot. But okay, so to look at the action potential itself, so again, we have our neuron, and let's say we're recording, we have a voltometer that is showing our membrane potential. But let's stick another electrode into the neuron and stimulate it with certain current, right? And this can trigger an action potential. The phases of the action potential are shown here. So our membrane potential on the y-axis, plotted against time. Several phases here. So here's our resting phase. This looks like it's sitting at, roughly negative 70 millivolts, somewhere between negative 60 and 70. But then the rising phase happens due to influx of influx of cations, causing a trigger. And what happens is once this membrane potential starts to shift towards the positive, you see this threshold value here. So before it reaches this, if you have a subtle influx of positive ions, it will go back to resting phase if it doesn't pass this threshold. So each cell has a specific resting threshold. Here it looks like roughly negative 50 millivolts in this example. And what happens then is you have this huge influx of sodium ions, right? That causes that rising phase. You have a bunch of cations that come into a negatively charged cell. It's going to shift the membrane potential, and as you can see, causes it to be at a positive value. This area here is known as the overshoot. That's the third phase. You start to have potassium moving in here, and then the potassium moving out as it's trying to correct that membrane potential. Then you have a massive flux of potassium moving in here, right? Sodium comes in. I keep saying this back and forth. My apologies, everybody. Sodium comes in, potassium goes out, okay? And that's how it balances this back, because you have a large influx of positive ions here, then a large outflow of positive ions here. It will overshoot the resting potential value, I'll call the undershoot, and then it rectifies itself, it gets back to the resting phase, all right? So after this, as you can then imagine, you have all kinds of sodium ions inside and a bunch of potassium ions outside. As we just showed, there's specific concentrations that should be inside and outside, and that's where the pumps come in, because they get turned on and they correct everything and they get the sodium and potassium back to their proper resting values. And that's the whole flux for this cell, all right? Why this is important is, well, I guess we'll get into that in a bit. So again, this happens once we're stimulating our dendritic ends. This happens all the way down the axon to the end of the terminal. How this happens is through the work of these channels all the way down the axon, and as stated before, if we have these highly specialized myelin sheaths, it will cause a much more rapid transmission of this axon potential down the axon. So as this shows, an unmyelinated axon has a flux of about 0.5 to 1 meter per second in terms of how quickly this will happen. So the axon potential happening here will trigger here, will trigger here, and on down. With these myelin sheaths, you don't have to move down the axon. It just jumps from node to node all the way down and shorts. Causes such an increase of speed that these myelinated axons can trigger or can work at a conduction rate of roughly 150 meters per second. All right, so a much faster type of transmission down these axons. So why this is important is due to the fact that the action potential will move all the way down, and as we'll see in the next couple slides, what then happens near the axon terminal is calcium gates are one of the last things to get activated down near the terminal, allowing for an influx of calcium into the system, and that actually will help for transmission of neurotransmitter out into the synapse in a process that we'll talk about in a moment. But show this just due to these electron micrographs here. So you can see each of these structures here is a vesicle. The darker ones are filled with neurotransmitter, the lighter ones are empty, and as you can see here and here, we actually have these bound to the membrane of the terminal and the pouring out of neurotransmitter out into the synapse. These active zones are where this is actually happening. So this is kind of the closest zoom up here is a bit backed out more, but this area that's dark here is where this is, a large area of this active release of neurotransmitter out into the synapse. Okay, and once again, why this is important. So, action potentials happen all the way down the axon. Here's a presynaptic nerve terminal, postsynaptic nerve terminal at the bottom. All the way down this axon, we have these action potentials. The synaptic vesicle can touch the membrane of this terminal. Influx of calcium triggered by this action potential allows for calcium to come in. And then this allows for these membranes to actually bind together, and then the release of neurotransmitter out into the synapse, all right? So, how this happens is through these snare proteins. Now, don't bother to memorize all the names of these, it's not important. Just know that there's snare proteins, all right? Snare proteins are bound by calcium. Calcium channel opens as a result of the axon potential reaching the terminal of an axon. They open, allow for influx of calcium, and as you can see here, so this yellow band is the membrane of a synaptic vesicle. Here we have the cell membrane, the plasma membrane of the axon terminus, and you can see that these snare proteins are embedded in this, in both membranes, and they actually come together and knit themselves together. And then what it will do is physically pull these membranes together, and then the membrane will part and it will open up and spill its cargo, the neurotransmitter, out into the synapse. And that's how neurotransmitter is released. That's how action potentials are coupled to neurotransmitter release. And once again, this process at the molecular level is facilitated by snare proteins. You don't need to remember the individual units, just that they are snare proteins. Calcium binds to them and allows for this stitching of the proteins together and then the pulling of the membranes together and apart, all in one spectacular motion. Okay? So, yeah, just to bring that back, or stitch these two figures together, here is here, right? And then once these snare proteins are activated by binding of calcium, pulls apart, and then this happens. Actually, we'll bind the membranes, cargo spills out, and then this empty vesicle can then be brought back up into the cytoplasm and recycled for the next round of filling with neurotransmitter. We'll talk about Tuesday, right? Okay, I think that's that for neurotransmission. Quick primer on that. How does that sound, everybody? Questions? Hopefully straightforward. Good. Please. Yes?
Is the chlorine pump ATP dependent?
So that one is not. There are pumps that are. For our purposes, it's kind of outside, but yeah. There are energy dependent chloride pumps. But yeah. Okay. They aren't all. Some of them use different for energy. Anyways. Good. Okay. So last bit, a number of agents affect neurotransmission. Here's the list. Botulinum, tetrodo, carby, marga, dendrochanotoxins, okay? And their origins here. So the most popular one everybody loves these days, well, it's kind of falling out of favor in certain areas. Clostridium botulinum, bacteria that develops a toxin but is able to block neurotransmitter release. As I'm sure everybody's heard here, this has been isolated, is now used in cosmetic procedure as Botox that can then freeze nerves and allow for you not to display wrinkles and all kinds of areas all over your body. But yeah, it's now used as a cosmetic and a horrible poison. It's been used punitively, but anyways, it is used as a cosmetic now due to the fact that it will chop up these snare proteins, right? So all of these different points on here are areas that the botulinum toxin can clip these snare proteins and not allow for neurotransmission to happen. Don't worry about beta ganglioside, heavy light chain, all this stuff. You're not going to be tested on. This is more the molecular underpinnings of this process. But just know that botulinum toxin is used to clip up snare proteins, and that will prevent synaptic transmission. That's what's important. Tetrodotoxin from the pufferfish. I'm sure some people enjoy sushi here. Hopefully you've tried this before. It sounds amazing, but this, if you're into sushi lore, this is one of the fish that has to be prepared in a very specific manner because the fact that this fish produces this tetrodotoxin and it is incredibly dangerous to consume. But the bottom line is that this blocks the sodium channel from activating, all right? Tetrodotoxin, sodium. Carbitoxin, margatoxin, dendrotoxin, all calcium channel blockers. The scorpion makes this one, the mamba snake makes this one. And so this blocks the potassium channel from working. As we said before, sodium comes in, potassium goes out during a normal axon potential. If you have one of these toxins on board and it blocks the potassium channel, it's going to shift your action potential to look something like this. You have a normally functioning sodium channel, but we blocked our potassium channel and it delays that inward flux, allowing for, that efflux of potassium, causing a delay in that rectification of that current of the charge within the cell, okay? So, carbatoxin, margatoxin, dendrotoxin, potassium. Conotoxin is used as a calcium channel blocker, isolated from a sea snail, and this is pretty amazing if you've ever seen one of these, nail its prey with this. This is really dangerous stuff, but whatever. Conotoxin block calcium channels, so they'll also block nerve transmission. Interestingly enough, this was investigated as an analgesic medication. The thought being, you have pain in a sensory nerve in the periphery, it communicates that up into central nervous system, that goes up into the brain, it says, I'm in pain. But if you're able to block this calcium channel, then it freezes that pain and you don't experience it. I don't know. Well, one interesting thing about it is there was an intrathecal administration of this, so if you've ever had one of those shots, you will always remember that. Getting it to block calcium channels, though, it makes sense on paper, but as an actual therapeutic, it's a little dodgy. But it can happen, I guess, is the bottom line. So the idea is that conotoxin can block calcium channels, and that's important for things we'll talk about later. But it has been explored as an analgesic. Just a bit of background on that. So that's really what I wanted to talk about today. Neurons are highly specialized cells that have this communication between them through synaptic transmission. This occurs through neurotransmitter release that is coupled to the action potential that moves down the entirety of that axon. Different molecules can disrupt this. Botulinum toxin, cuts snare proteins, tetrodo blocks sodium, carbon marga, dendroto blocks potassium, and conotoxin for calcium. So quiz today, it's only on the PKPD lecture, so today